Hi everyone, my name is Letícia. I am graduated in biology from the Federal University of Juiz de Fora, and I am currently enrolled in the graduate program at the Federal University of Juiz de Fora, pursuing a master's degree. I am supervised by Dr. Vanessa Cordeiro, and my project is entitled Trichosporum spp. What is new? The genus Trichosporum comprises several species of fungi belonging to the phylum Basidiomycota. They are uracid positive, non-encapsulated, and yeast-like organisms. These fungi are widely distributed in the environment and can be found in soil, water, and as part of the microbiota. The pathogenicity of this species of fungus is directly associated with virulence factors that facilitated fungal invasion and establishment within host tissues. Among the pathogenic species, Trichosporum acai and Trichosporum asteroides are responsible for the majority of trichosporonosis cases, representing a growing concern for public health. The clinical manifestations of TSI infections vary according to the affected organ and may include fever, cerebral and hepatic abscesses, fungemia, and respiratory or urinary tract infections. This fungus is opportunistic in nature and can cause disseminated and invasive infections, particularly in immunocompromised individuals, with mortality rates ranging from 30% to 90%. Despite the growing relevance of the genus Trichosporum in medical practice, the treatment of infections caused by this species still faces significant challenges. The scarcity of data on the efficacy of antifungal agents against this clinically important species limits therapeutic options. Recent studies highlight several antifungal resistance mechanisms, both molecular and non-molecular. Among the most common molecular mechanisms are mutations in ERG genes involved in ergosterone biosynthesis and the overexpression of efflux pumps mediated by the MDR1, CDR1, and CDR2 genes, mainly associated with azole resistance. Because of this, my study is going to evaluate physiological and molecular characteristics related to resistance to the main antimicrobial agents in Trichosporum spp. Isolated obtained by Microbiology Service of the Cortez Vilela Laboratory, located in Juiz de Fora, Minas Gerais, Brazil. The specific objectives are: evaluate the clinical and epidemiological aspects of community and hospitalized individuals with Trichosporum spp. Infections treated between January 2020 and December 2023. Determine the antifungal susceptibility profile to clinical relevant antifungal agents. Assess the presence of antifungal resistant genes among clinical isolates, and to establish correlations between the presence of genetic markers related to antifungal resistance and the antifungal susceptibility profile of the isolated fungal strains. So, This is a descriptive, experimental, and cross-sectional study consisting of a retrospective analysis of the years 2020 and 2023, based on electronic medical records of hospitalized and community patients with Trichospora spp isolates attended by a clinical microbiology service in Juiz de Fora, Minas Gerais, Brazil. This study was conducted by participants' consultation and informed consent in accordance with a project approved by the Research Ethics Committee involving human subjects of the Federal University of Juiz de Fora, under CAAE informed in the presentation. This experimental design of this project includes cultures that will be prepared from the isolated obtained from the samples, and the yeast identification is going to be by the Vitex system. The antifungal susceptibility testing will be carried out using the disk diffusion method, employing the main antifungal agents used in clinical practice. The DNA extraction and quantification will be performed from the yeast isolated cultures, and the extracted DNA will be used for PCR to identify antifungal resistant genes of medical relevance. The results will be analyzed to understand the expression of these genes in Trichospora species, isolated from clinical specimens. Finally, patient data will also be evaluated based on electronic medical records to determine clinical and epidemiological factors. The findings in this project are expected to provide support for epidemiological surveillance practices, rational antifungal selection, and the development of laboratory protocols that reduce diagnostic time and promote better clinical outcomes. Thank you.